everyone. So in this chapter also we are going to continue with our topic which is with our chapter which is photosynthesis in higher plants. So till now we have covered a great portion and wait a minute. Okay. So we are going to continue with our okay ये पढ़ लिया था. Splitting of water was also done. This was also done. Chemiosmotic hypothesis was also done. And yes, we will be discussing these points. So let's get into the video. Okay, so what is it saying? It is saying that as the electrons move through the photosystems, okay, protons are transported across the membrane. There is this transportation of protons. Like there was a difference. Like inside the membrane, it was like inside the membrane. It was like there was accumulation of protons. So there was a pressure gradient. There was a gradient. Sorry, there was a gradient as there was difference in the number of protons. So there was a gradient, and due to which what we did, that protons are transported across the membrane. There was movement of protons from inside to outside the membrane, which is from lumen to stroma. Okay, next is saying that this happens because the primary acceptor of the electron has located towards the outer side of the membrane and transfers its electron not to an electron carrier but to the H carrier. So the molecules removes a proton from the stroma and transports an electron, while this molecule passes on its electron to electron carrier on the inner side of the membrane, and the protons are therefore released in the inner side or the lumen of the membrane. Next, what happens? The NADP reductase enzyme is located. It is located. We have an enzyme called as NADP reductase. And what's the role of the enzyme? The role of the enzyme is that it is an enzyme which is located in the stroma side of the membrane, and along with the electrons that comes from the acceptor of the electron of ps1, and these protons are necessary, like they are needed always for the reduction of NADP to NADPH and H positive. So there will be NADP reduction and it will form NADP H plus H positive. Okay, and these protons are also removed from the stroma. So within the chloroplast, the protons in the stroma decreases in the number, and while in the lumen there is accumulation. So accumulation of protons, and this. So what happened? This is written only here. You don't need to even read all of this. This is telling that what actually happened. So within the chloroplast there was protons in the stroma, and what happens inside the lumen, there was the accumulation of protons. And if there is a difference between the protons accumulation, like inside the membrane which is lumen, it is accumulated, and outside there is like it is negligible. It was negligible. So it makes a gradient and proton gradient across the thylakoid membrane, as well as the measure decrease in the pH. So therefore, this gradient is very important because it is the breakdown of the grade of this gradient that leads to the synthesis of ATP. So only because of the only because of the gradient this all happened, because when the gradient breakdowns when the gradient breaks down, it synthesizes ATP. So here we have a great role of gradient also, and because of gradient this was done. Okay, this all was done. And the next it is written that the ATP synthase enzyme consists of two parts only. Is CF node embedded in thylakoid membrane and forms a transmembrable channel that carries out facilitated diffusion of proton across the membrane, and other portion is CF1, and which protrudes the outer side of the thylakoid membrane and of the side of that faces the stroma, and the breakdown of gradient which provides us energy to cause a conformational change in the CF1 particle of the ATP synthase, which makes enzyme synthesize what? Our energy, which is ATP. Chemoosmosis requires a membrane, a proton pump which will pump the protons, okay, from inside to outside, a proton gradient, or we can say ATP synthase. And energy is pumped proton across the membrane and to create a gradient or high concentration of protons within the thylakoid lumen. And ATP synthase has a channel that allows the diffusion of proton back across the membrane. This releases enough energy to activate this enzyme, and which leads to the formation of ADP. Okay, next we are going to talk about that how, where are these used. Like we have synthesized the ATP by making a pressure proton gradient, proton gradient. We have made a proton gradient, and by the help of proton gradient there was the breakdown of, when there is breakdown of proton gradient, then it leads to the synthesis of what? Our ATP. So we have made the ATP. Now the question comes that where we need to use it, like where we need to use it, huh? So we are going to study in this para. What is it written? It is written that we learned that the products of light reaction are ATP, NADPH, and O2. Of these, O2 diffuse out of the chloroplast, whereas ATP and NADPH are used to derive the process of leading for the synthesis of food, okay, and more accurately sugars. Okay, and this is biosynthetic phase. This is biosynthetic phase of photosynthesis. This process does not directly depends on the light, because it is dependent on the product, and which is dependent on the product is called as dark reaction. पढ़ा था ना? Dark reaction. What was dark reaction? Look, light reaction और dark reaction में यही difference है, that light reaction is that reaction in which we need the light energy. We need the sunlight energy. We need the light energy, and the light falls on the membrane. Like directly, it is directly dependent on the light energy. And dark reaction is that reaction in which it is not directly dependent, but somehow depends on the product of this light reaction. Okay, depends on the products of the light reaction. So indirectly it is also dependent on the sunlight, because it is dependent on light reaction means it is indirectly dependent on the presence of light. Okay, presence of light, but dependent on the products of light reaction, ATP and NADPH, besides CO2 and H2O. Now we are going to talk about, yes, let's now see how the ATP and NADPH are used in the biosynthetic phase. Okay, so we saw earlier that CO2 is combined with H2O to produce CH2ON, or sugars, we can say sugar C12H24O11. Okay, and it was interested to the scientists to find out that how this reaction proceeds, or how the CO2 is taken, and how this happens. Okay, so we have something for it. We have a scientist named Melvin Kelvin, His studies led to the discovery that the first CO2 fixation product was 3 carbon organic acid, which was 3 carbon organic acid, and the product was identified as, later it was identified as 3 phosphoglyceric acid, we can say PGA. So the cycle which he gave was a pathway which is Kelvin cycle. Kelvin cycle given by whom? Melvin Kelvin. And discovered that the first product of CO2 fixation was what? 3 carbon organic acid, which was 3 phosphoglyceric phosphoglyceric acid. Done. Let's talk about next it is written, scientists also try to know whether all the plants have PG as a first product that we see in the, like here we read that the first product of CO2 fixation was 3 phosphoglyceric acid, but later the scientists were like very curious to know whether the same happens to the plant or not, or something else. So the first product of CO2 fixation, or whether any other product was formed in the plants. So experiments were conducted to find the answer of their curiosity. And okay, and here it is written that experiments conducted over a wider range of plants led to the discovery of another group of plants where the first stable product was again an organic acid, but not that which we discussed in the later previous one, but different from that. And it was oxaloacetic acid. It was oxaloacetic acid. Okay.